Hello everyone. In the last video, I have discussed about single radial immunodiffusion technique, which is also called as Mancini test. The technique is based upon precipitation reaction, which is nothing but the antigen-antibody interaction. Okay. In case of precipitation, soluble antigen, okay, which is soluble or non-particulate, react with its antibody to produce visible precipitin complex. Okay, now this precipitin precipitate out from the reaction mixture and become visible as a faint white line. Okay, so this is precipitation reaction and it only occur in presence of electrolyte like NaCl and at optimum temperature and at optimum pH. Okay, so we can define precipitation as antigen-antibody interaction in which soluble or non-particulate antigen combined with its antibody to produce visible precipitin complex or visible lattice, and this reaction only occur in presence of electrolyte like sodium chloride and at optimum temperature and pH. Now the precipitation reaction can occur in both, that means in liquid medium or in gel medium. If the precipitation occur in the liquid medium, then the well-known example of this type of reaction is precipitin ring test. While when the precipitation reaction occur in the gel medium, it is called as diffusion. Okay, and the three important examples of the diffusion are: number one, single radial immunodiffusion; then double immunodiffusion; and third is immunoelectrophoresis. Out of which the single radial immunodiffusion, it is the technique in which in the gel we add antibody, so the antibody remains stationary while antigen move towards antibody. So only antigen is moving, so it is called as single. Antigen move radially, so it is called as radial. And as antigen and antibody involved, so it is called as immunodiffusion technique. Okay. Another name of this technique is Mancini test. Similarly, the next example of gel medium diffusion is double immunodiffusion. So now in this video, I'm going to focus on this technique, double immunodiffusion. Okay, so the name itself suggests it is the type of a double immunodiffusion. That means now, unlike single radial immunodiffusion, in this technique, both antigen and antibody can diffuse freely towards one another in the gel. Okay, so for example, suppose this is the plate, glass plate, and on which we have poured molten agarose. And now after solidification, we made two wells in this agar or agarose with the help of well puncture. And in the one well, we add antigen and the other well, we add antibody. Now, in case of double immunodiffusion, both antigen and antibody can diffuse towards one another. Both are free, both are mobile inside the gel. Okay, so it is called as double immunodiffusion. The technique of double immunodiffusion was first time discovered in 1948 by Ouchterlony. Okay, so this Ouchterlony first time report the technique of double immunodiffusion in 1948. So this technique is also called as Ouchterlony technique. Okay, now the technique is mainly used for qualitative estimation of antigen or antibody. Remember that single radial immunodiffusion is mainly used for quantitative estimation of antigen or antibody. That means you can find out the concentration of antigen or antibody from the sample by using single radial immunodiffusion. But now the double immunodiffusion is mainly a qualitative test. In this type of test, you can only detect presence or absence of antigen or antibody from the sample. Another application of this technique is that you can compare different antigens. Okay, so the technique have comparative application. You can compare different antigen present in different sample with the help of this technique. Okay, and you can also determine purity of the antigen or antibody by this technique. Okay, if we consider principle of double immunodiffusion, in that case we can say in this technique both antigen and antibody are free. Okay, they can diffuse with towards one another radially in the gel. Okay, and when they meet at zone of equivalence, precipitin complex is produced. And when this complex is produced at the zone of equivalence, which is present between antigen and antibody, a white precipitation line develop. Okay, so when this antigen move towards antibody at zone of equivalence, we can see a white precipitation complex or lattice. Okay, so the appearance of this type of a line is the indication of compatibility of antigen towards antibody. That means both this antigen and antibody are specific towards each other. The line of precipitation only appear at zone of equivalence. In my last video of precipitation, I have discussed in detail about the principle of precipitation, in which we found that there are three different zones when antigen and antibody move towards one another. Number one is pre-zone. So in pre-zone, antibody are in excess, while in case of post-zone, antigen are in excess. In both these cases, precipitation reaction do not occur. The reaction only occur when antigen and antibody are present at equal concentration, and it is called as zone of equivalence. When zone of equivalence reach in the gel, you can find precipitin or lattice complex, and at this point the precipitation reaction occur. Okay, now if you want to perform this precipitation reaction in the lab, you should need some requirements. Okay, so now we are going to make list of the requirement. Number one is agarose, or you can use agar or polyacrylamide as a solidification medium. Okay, so powder of agarose or agar or polyacrylamide. Then the second is assay buffer. All the type of antigen antibody interaction like agglutination, precipitation are carried out in assay buffers, because these are proteins. Antigen and antibody are protein, and they require optimum pH for their activity. And during the reaction, if pH get fluctuated, in that case chances of denaturation of antibody increases. And to avoid this, we should keep pH of the reaction medium constant, and for that we use assay buffer. Okay, then the third requirement is antigens. Okay, to perform double immunodiffusion, you will going to need different antigens, okay, which you want to detect from the sample. Then the next is antibodies. Okay, antibodies or antiserum which is specific for this antigen. Okay, and another requirement for this is instrument like incubator, some glasswares like conical flask, measuring cylinder, and well puncture. Okay, we use this well puncture to make well in the gel. Okay, so all these are the important requirements that one should need for performing double immunodiffusion technique. Now we're going to discuss about procedure of the technique. Similar to that of the single radial immunodiffusion, in case of double immunodiffusion, the first step is preparation of gel. Okay, you can prepare gel of agarose or agar or polyacrylamide by dissolving 1% or 1 gram this powder agarose in 100 ml buffer. Okay, so you have to just weigh 1 gram agarose, and then you are going to add that agarose in 100 ml buffer, assay buffer, and initially the solution will going to appear the blur, and so that you have to boil it to dissolve. Okay, so after mixing 1 gram agarose in 100 ml buffer, boiling will be done, and during the boiling, the agarose will going to get dissolved in the buffer, so that you will going to get clear gel. Okay, so after boiling, clear gel or clear solution will going to appear, and after that, after boiling, the third step is allow gel to cool up to 55 degrees Celsius, 55 to 60 degrees Celsius. Okay, that means you have to decrease the temperature of this gel up to 55 degrees Celsius, and after that, the next step is addition of antibody. Okay, we know that and antigen, antibody and antigen in well. We know that in case of single radial immunodiffusion, we generally add antibody in the gel, but now in this case, after cooling the gel to 55 degrees Celsius, we will going to pour this gel onto the glass plate, and after pouring the gel, we will going to make well in the gel with the help of well puncture, and finally we will going to add antibody and antigen in different wells in this gel. Okay, and after addition of the antigen and antibody, next step is incubation of the glass plate at 37 degrees Celsius for 24 to 48 hours. Okay, generally this incubation will going to carry out in incubator. Okay, at 37 degrees Celsius in moist chamber. Okay, I have discussed how to prepare moist chamber in the lab with the help of cotton and glycerol ball. So we have to just prepare cotton ball, and then this cotton ball soaked in the glycerol will going to maintain the moisture of the incubation chamber, and this moisture will be very important for reaction of antigen and antibody. So at the time of incubation, the moist condition is necessary, and after these steps, the last will be the observation and interpretation of the result. Okay, so three different type of precipitation line can appear after this procedure. Okay, so first is preparation of gel. For example, I prepare a gel, 1% agarose gel, and I dissolve it in the buffer with the help of boiling, and then after cooling I pour the agarose gel onto the glass plates, clean, grease-free glass plates. Okay, now after that I am going to make wells like this in the gel medium with the help of well puncture. And after making such wells, the next step is addition of antibody and antigen. For example, in the upper well, I am going to add antibody, while in case of all the lower wells, I am going to add antigens. And after that, we are going to incubate all the plate at 37 degrees Celsius for 24 to 48 hours in moist condition inside the incubator. And the next day, we are going to see the results. Three different types of precipitation line can be obtained. Okay, so we're going to discuss each result one by one. In case if first we add antibody A in this well, we're going to show antibody by capital letter, and if we add antigen small a in both these wells. Okay, so in the first well I add antigen small a, and in the second well I also add antigen small a. Now this capital A antibody is specific for this small a antigen, so that once this antigen diffuse towards antibody and antibody also going to move towards antigen, at zone of equivalence they will going to produce precipitation line like this. So this is the line of precipitation which is formed between antibody A and antigen A. Similarly, same line will going to appear in between these two, antibody and antigen, so that if we get V-shaped or arc-shaped precipitation line in between one antibody and two antigen, then we can say that these two antigens are identical. Okay, so with the help of this type of results, we can conclude that the two antigen which is loaded into the two wells are identical with one another. They have same epitope, so that similar lines are obtained. The first line is the result of reaction of antibody A with antigen A, and this line is also going to produce due to the antibody A antigen A reaction. Now in the second case, if we add antibody A and B, mixture of antibody, okay, I add mixture of antibody A and B in the first well, and in the next two well I add antigen A and antigen B separately. In that case, what will going to happen? This antibody A will going to react with antigen A, and they will going to produce precipitation line like this. Okay, and similarly this antibody B will going to react with antigen B, and it will going to produce precipitation line like this. So if we get such type of cross precipitation line, it is also called as crisscross precipitation line, then we can say that both the type of antigen in the well are non-identical. Okay, because the cross is produced, and this cross is only due to the interaction of different antibody with a different antigen. Okay, lines are crossed to one another, so no one can conclude that both these antigens are non-identical, they are non-similar antigens. And in case of third or last, if we add antibody like A1 and A2, okay, this is the antibody, single type of antibody which is specific for two epitope present on the antigen. Okay, so now you have to focus here. I have used single antibody. For example, this is the antibody, Y-shaped antibody, and now the first antigen binding site of this antibody is specific for A1 antigen, and second antigen binding site of the antibody is specific for A2 antigen, because antibody is bivalent, it can bind to two antigen. Okay, so now this antibody is called as A1 A2 because it have two sites for binding on the antigen, that is A1 epitope and A2 epitope binding site on the same antibody. Now if I add one antigen into this well, and this antigen have epitope A1 and A3 type, and in the second well I add antigen which have epitope A1 and A2 type. Okay, listen carefully now. Now this A1 antibody will going to react with A1 antigen, and A2 portion of the antibody will going to react with A2 epitope of the antigen. So it will going to produce line like this, quite long line. Okay. Now in the second case, the A1 antibody portion will going to react with A1 epitope of the antigen, which will going to produce precipitation line like this. But now this A2 portion of the antibody do not have binding site on the antigen, because this antigen contain A3 type of epitope. This A3 epitope is non-specific for A2 binding site of the antibody, so that this A2 binding site of the antibody remain free, and this A2 binding site will not going to bind to A3 antigen, so that this second line will going to remain short. Okay, this is small line and this is quite longer line of precipitation, so that a Y-shaped precipitation line produce. And this Y-shaped precipitation line indicate that both this type of antigen A1 and A3 and A1 and A2 are partially identical. Okay, so now this portion of the precipitation line is the result of combination of A1 antibody with A1 antigen and A2 portion of the antibody with A2 epitope of antigen. So this longer line is due to A1, A1, A2, A2 reaction, while the smaller precipitation line is due to the reaction of A1 epitope binding site of the antibody with A1 epitope. This A3 epitope will going to remain free, so that this line appears small. Okay, now this Y-shaped precipitation line indicate that both the antigen are partially identical, because they have A1 epitope which is similar, but they also have A2 and A3 epitope which is dissimilar. So with the help of the double immunodiffusion, you can easily compare different antigen. For example, if I get arc or V-shaped precipitation line, I can conclude that both the antigens are identical with one another. They have same epitope on their surface. If I get cross precipitation line, in that case I can declare that, or I can conclude that both the type of antigens are different from one another. They are non-identical. And if I get Y-shaped precipitation line, this is because two epitope binding site of antibody can bind to both the epitope of one antigen and only one epitope of another antigen, so that this Y-shaped line is appear. This extra portion of precipitation line is called as spur, and this spur happened due to partially identical antigen. So we can conclude that this double immunodiffusion or Ouchterlony technique is the effective qualitative and sometimes quantitative antigen-antibody interaction. Thank you.